This is a prepared slide of a pig embryo where we can see developing tissues and organs that originally came from precursor stem cells. Now, embryology is not the topic of this video, connective tissues are. This slide just gives us a clue as to how connective tissues are developmentally related, and that has to do with the mesenchyme. Now we're zoomed into an area containing mesenchyme, which is derived from the mesoderm. It is the link to most all components of connective tissues as well as other organs and tissues. So it's a great example of what links this very diverse group together. Here we have a bunch of unspecialized cells in here that have potential to differentiate into many different typical cells in adult connective tissues. You can see some extracellular parts that are a bit more string-like. The oval shapes right here, all of these, those are nuclei of cells, and you can see a bit of the cytoplasm. They can differentiate in most cells of the connective tissues. Without getting too technical, such as excluding blood cells, it is the link between connective tissues from a developmental standpoint where we see cells and an extracellular matrix. Let's jump in and explore the nature of connective tissues. Connective tissues are largely support tissues, meaning they anchor and join other tissues together. But that's a bit of an oversimplification because it's true for most, but not all connective tissues, as they have other functions. Here's a table of various different connective tissues. At first glance, you may think, what is adipose, that is fat, how's that connected to cartilage, and how's that connected to blood? And what they all have in common is they are derived from embryonic tissues in the mesoderm. The extracellular matrix is also very important. Unlike in epithelial tissues, which are mostly made up of cells, connective tissue contain a lot more important things outside of just the cells, namely protein fibers and ground substances. Now, we're going to divide this video into two parts. The first half is dedicated to connective tissue proper referring to authentic or more genuine. The second half, we're going to deal with specialized connective tissues. Technically, we can further subdivide these into support connective tissues and fluid connective tissues, but they're classically considered specialized connective tissues. Now, the ones highlighted here in yellow are the ones that we're going to look at in this video. The ones that are not highlighted, we're not going to look at because they're typically not tested in intro histology labs. So they're included in this table for completeness. As we go through each and every one of these tissue samples, be sure you're paying attention to both the cells and the extracellular matrix contained within those tissues. Cells contained within connective tissues can be fairly diverse. Fibroblasts and adipocytes are among the easiest to spot. Fibroblasts help build and maintain the extracellular matrix, the ECM. They have an elongated nuclei, so they're fairly easy to spot. Adipocytes, they store excess energy in the form of fat, so they're useful in that respect, but they also help create a cushion and add to insulation when working together. The other cells here, namely the macrophages, mast cells, and other leukocytes, are involved in the immune response. They can be a bit harder to spot and identify. Mesenchymal cells can also be present, and they can differentiate in other cells when needed. The ground substance here is comprised of a number of proteins and carbohydrates called glycosaminoglycans. So whenever you look at a sample, you'll see cells and you'll see fibers, and you'll see this, what appears to be an empty or colored space, such as here, here, or over here, all around this image. That's where our ground substance is located. And it's this watery-like background that has some viscosity and allows for diffusion of nutrients, as well as the free movement of some cells. Think about how our immune cells need to move around and attack foreign invaders, or they signal repair to other tissues. They're moving through this ground substance. Lastly, there are a number of various fibers that help bind things together. Namely, collagen fibers are relatively thick, and they're the ones that bind things together. Here we have a collagen fiber, it's thicker. There's another collagen fiber, it's thick. So think about grabbing and pulling your skin. These fibers only allow you to pull your skin so far. So depending on the source, there are about 28 different types of collagen, with type 1 collagen being the most notable. Elastic fibers are thinner and more narrow, and they allow for stretching and retraction. So here, these green fibers right here, they're going in a bunch of different directions. They're a lot thinner. So think about them as like a rubber band. Now, if you were to pull your skin and let go, these fibers would be responsible for the quick snapping back of your skin once you release it. Reticular fibers are another one. They're actually type 3 collagen fibers. They're thin and branched, so like over here, these blue ones, kind of like a spider web. So they help form this framework that can help contain certain tissues. Now, the terms loose and dense connective tissues, that just has to deal with how the fibers are organized, as being bundled together, being dense, or not being bundled together as loose. So all connective tissues, and these connective tissues proper, have these same elements to a certain degree. The makeup and the number of cells and fiber types is how we differentiate their names and what will make sense as we go along through each of the tissue type. Now, let's look at our first two connective tissue samples under loose connective tissue, areolar and adipose. Areolar tissues act as a filler space between other organs and are a component of serous and mucous membranes. Most of the nuclei that we're going to see are from cells known as fibroblasts. They have a granular and elongated looking nucleolus and nucleus. They stain well with H&E staining, which this is over here. Note though, as opposed to our fibroblast over here that has these cellular extensions, those don't really show up very well in an H&E staining. Instead, we only see the nucleus and nucleoli. So here's a good example. Elongated nucleus, dark staining nucleus and nucleoli. Here we have another one, and there's a good one over there too as well. So depending on the sample and location, we could see nucleoli of adipocytes as well as other white blood cells, but it's safe to say for an introductory lab, these are all nuclei of fibroblasts. The other two structures we see are different fiber types. This one right here is our collagen fiber. It's relatively thicker and stains lighter. They have high tensile strength, but are not elastic. The thinner and darker staining fibers, like this one over here, are our elastic fibers. They allow for more stretching and subsequent retraction, kind of like a rubber band. Now, areolar tissues are loose connective tissues. It might look fairly dense in certain areas, perhaps, but they're technically classified this way because these fibers do not occur in bundles like we will see later. Here we're looking at adipose tissue, more specifically white adipose, which is most abundant in adults. There is a single large lipid droplet here in the middle. In brown fat, or as known as baby fat, there will be multiple droplets inside of this cell. Simply put, this slide is what we commonly think about when we think about fat. Here is our adipocyte. Draw around it. So because there's this single large lipid droplet here, it pushes the nucleus out towards the outer part of the cell. Really, it's still inside of it, and we can see a bit of the plasma membrane surrounding it here. These cells largely contribute to lipid molecule storage, thus you can think about them as energy storage cells. However, they also contribute to insulation from cold temperatures and providing cushion from mechanical injury. We commonly, or at least mainly, think about these as being just below the skin, but adipose is located around internal organs and in serous membranes to help protect them as well. Note that with most stains in fixing the slides with standard protocols, it actually dissolves the fat. So we're really just looking at an empty space on this slide right here with the plasma membrane and a nucleus. Also note that these cells are really 3D and spherical, so based upon where we cut this thin slice through the specimen, you might not actually see a nucleus in some cases, and it just looks like a circle where we just can see the plasma membrane. One last thing that we don't see much of in this slide is fibers. That is because adipocytes are most commonly found clumped together or surrounded by other components. For that reason, some authors and textbooks might consider adipose as a specialized connective tissue, but we're going to include it here because it really follows how we name connective tissue proper tissue samples. So those were our loose connective tissues. Now we're moving into our dense connective tissues, starting with dense regular connective tissue at 100 power. Now with our typical H&E stain, we see these nuclei that are staining dark purple, And we also have numerous bundles of collagen fibers that are densely packed together and stained a little bit more pink. This type of tissue is found in tendons and ligaments, where we need a lot of tensile resistance. Think about lifting weights and the tension that is placed on the anchored bone to get the movement, or in a ligament resisting movement, like if you were to try and bend your fingers at the joints. Now, let's zoom in a bit and see what more we can see. So now we're at 400 power, and aside from getting a closer up view, we can't see much of anything else. We can see the fibroblasts, or rather the nuclei, that are these purple staining dots that are circular to oval in shape. And we can see our collagen fibers, which are orientated roughly left to right and have this wavy-like appearance. Now, these fibers are bundled together and they are poorly vascularized, so they don't get a whole lot of blood flow. This tissue can appear similar to muscle tissue, but it lacks perpendicular striations that we'll see later. In the typical H&E stained specimen, this is what we see are a lot of fibroblasts and a lot of collagen fibers, and really not a whole lot of anything else that we would see in our connective tissue proper. Dense irregular connective tissue. So this is skin at 100 power. We see the epidermal lining right here containing epithelial tissues. Down in here, we're in the dermis. We can see some glands, some blood vessels, but for the most part, we're looking at this region right here, which is dense irregular connective tissue. There's a lot of different parts to it, but for the most part, it has this wavy-like appearance. Now we're going down further, and we can see some adipose tissue. We're now in the hypodermis, so there's a lot of fat there. But if we go back up into our dermis right here and zoom in, now we're zoomed into 400 power and we're focusing in. We can see these dots, those are our nuclei, and the rest of this is collagen fibers in bundles, and it has this rather wavy-like appearance to it. So now we're at 400 power in the dermis, but do note that this is actually a different specimen in that the fibroblasts, the nuclei are right here, here, and here. Note that they are circular to oval in shape, but they don't stain quite as good as the last slide. But where this slide is a lot better at demonstrating is the collagen fibers. And do note that the collagen fibers are densely bundled together, therefore that's dense. They are irregular because they're going in multiple different directions, whether it's more longitudinal or maybe more obliquely or more cross-sectionally, all over the place. You can see that it doesn't really have a specific pattern to it, thus the irregular part to it. So just like in dense regular connective tissue, we mainly see collagen and fibroblasts, but do note that we can observe some of these other cells and also other fibers, typically with different stains though. Quick review of connective tissues. They're largely support tissues. They have the same embryological origin, and they're all about cells, mainly the fibroblasts, but others that are focused in on maintaining and building the extracellular matrix. Those were our connective tissue proper tissues. Now we're going to look at the specialized connective tissues, which are over here in our table. Note that instead of fibroblasts, we have a specialized cell that creates this extracellular matrix that's quite a bit more diverse and different from connective tissue proper. We'll model hyaline cartilage for cartilage, but do note that there are other types of cartilage. We'll also do bone, and then we'll end this video on blood. So there are 3 different types of cartilage, with hyaline cartilage being the most abundant in humans, so we'll focus on that. Cartilage has a lot of functions such as protecting bones from abrasion inside of joints and distributing mechanical loads. Here we see it in the trachea, also known as the windpipe, where it provides structural support to prevent the trachea from collapsing. So it's pretty important since we need air to breathe. So it helps maintain the shape and keeps this airway open. If we cut right through the trachea, that's what we have here on this slide. Now we'll zoom in. Note that these tracheal cartilage rings are rather C-shaped overall, and depending on where the cut is, they might appear less C-shaped, or you might only see part of it. In the middle right here, this is our airway. That's what we want to keep open so we can get air in and out. So it's very, very important. So let's zoom in right here into the cartilage and explore some of the structures and functions of this tissue. Here we're at 400 power. The main cells of cartilage are chondrocytes. Embryologically, they are derived from mesenchymal cells, so they're very similar to fibroblasts, although they're a bit more specialized. They actually start off as chondroblasts, which secrete a matrix that ultimately surrounds them. So in this sample, here we have our main cells. Those are the chondrocytes. Here's one right here. And it's contained within a space or a cavity that we refer to as the lacuna. They can occur by themselves or they can occur in clusters. The extracellular matrix is comprised of proteoglycans and has this gel-like consistency to it and is glassy in appearance. Now, note that it does have collagen fibers within it, although they're not easy to see and require different techniques to visualize them. Here we see the perichondrium, peri meaning around, or it surrounds the cartilage. This is actually dense, irregular connective tissue. Now, it's interesting to note that cartilage itself is avascular, meaning it does not contain blood vessels. Instead, it relies on the diffusion of blood from the blood vessels over here in the perichondrium. So cartilage in itself has a very limited ability to heal itself. That is why people who have injuries to their joints oftentimes have a difficult time recovering and might develop degenerative joint disease and might ultimately need a joint replacement surgery. So cartilage is a specialized connective tissue and it follows our standard description of what it is. There are cells, the chondrocytes, that help build or maintain the extracellular matrix, which is comprised of fibers and ground substances. So it is a specialization that has to do with the cell types and the ground substance. For bone, also known as osseous tissue, there are some important structures. First, though, the sample that we're looking at is compact bone. Compact bone, right here to here, that's more towards the outer part of the bone versus spongy bone, which is located more towards the middle of the bone, the organ itself, that is. So the primary cells are the osteocytes, and they're contained within spaces called the lacuna, just like cartilage. So each and every one of these small little dots right here, as well as down here, that's going to be where our lacuna are, as well as our osteocytes. Note that they have canaliculi, which are these small canals that are radiating around them and through them so they can communicate to other cells. Now the ECM in this case, the extracellular matrix, is comprised of collagen fibers that are surrounded and embedded in hydroxyapatite, which is the strong mineralized substance. It's fairly organized, and thus with this diagram, we have a lot of terminology. We're just going to identify a few different things. First, the osteon. Right here's one. There's another one over there. So these osteons, they are the structural and functional unit. It's essentially how bone tissue is organized to have its strength. They are cylinder-shaped and arranged in parallel lines to the stress of the bone. Next, we have our central canal and perforating canals. So bone is fairly vascularized. So there are a number of blood vessels and nerves inside of it. So we need canals for them to run. The central canals, right here, there's a central canal right there, and another one over there. So those are running in the middle of our osteons versus our perforating canals. Perforating canal there. They're running perpendicular to those osteons. So they're both really important and essential. So now let's look at what bone tissue looks like up close. Now here we are viewing at 400 power a ground bone specimen. It is a thin slice of bone in which the organic material has been removed and then a dark dye has been used to visualize the smaller canals and structures. So each one of these black circular looking dots, those are our osteocytes. And the space that they're contained within is known as the lacuna. So you should be able to see some similarities between bone and cartilage. We can also see these tiny little canals or canaliculi that allow for communication and nutrient exchange between different osteocytes. Now, here's a roughly circular outline of an osteon. You might be able to see slight or subtle color difference inside and outside of the osteon, although it is very, very subtle. Now, note that in living bone, there are a lot of blood vessels and nerves. With this ground bone preparation, the non-osseous contents have been lost, so we don't see them. As such, there are no blood vessels or nerves contained within this slide that we can visualize at least. However, there might be a little bit of bone dust, like right here, those black specks right there. So it's more of an artifact to the preparation of the slide preparation. So right here we have our central, also known as Haversian canal, that contains our blood vessels and nerves, and they're running within the middle of the osteon there. Now, the opposite is true with our perforating canals, although we're going to have to look at a different spot on this slide to be able to see those canals. So now we move to another area on the slide in which we can see the outline of two osteons. There's roughly an osteon right here, and then there's another osteon right there. So therefore, here in the middle, we have our central canals that are running with the osteons, containing blood vessels and nerves. And therefore, running perpendicular to those, we have another canal known as our perforating, aka Volkmann's canal. So, in summary, bone is a specialized connective tissue. The main cells being osteocytes, although there are a couple others that are not mentioned here, and the extracellular matrix, which is comprised of embedded collagen fibers, although not easily seen in the slide preparation, and those fibers are surrounded by a mineralized hydroxyapatite with the mentioned patterns, and it has both blood and nerve supply. So again, specialized cells and specialized extracellular matrix. Lastly, we have blood. Now, blood functions to transport nutrients and waste between organs and tissues, among many other functions. And it may seem odd at first to consider blood a tissue, but it's comprised of two things. First, formed elements, that's our cells and platelets. And second, we have our extracellular matrix, that is our blood plasma. So if we took a vial of blood and we centrifuged it, that is, spun it in circles at really high speed, we would end up seeing layers that look like this. At the very top, we have the least dense part, that's our blood plasma, our extracellular matrix. It is there to suspend the blood cells and allow them to circulate. It's mostly made up of water, but there are a number of important proteins, electrolytes, hormones, among other nutrients. The hematocrit here at the bottom, it's the densest. That's where we see our erythrocytes, our red blood cells. Now, they primarily function with transporting oxygen, and their shape's a little bit unusual. Like over here, it kind of looks like a donut, actually. There's not an actual hole here in the middle like there is in a donut, but it's really thin and it appears transparent. Depending on the angle, like some of these over here, you don't see that hole, but there is technically a spot where there's something missing. And that thing that's missing is the nucleus, because red blood cells, they used to have nuclei, but they lost them during development of the cell. Then we go to our buffy coat right here in the middle. This part is very, very little in overall blood volume, about 1% or so. Whenever you see these tiny little specks, those are our thrombocytes, also known as our platelets, and they function with blood clotting. And the rest of the buffy coat are our white blood cells, our leukocytes. Now, they have nuclei that appear more purple or dark blue, as we typically see in most of our histology slides. And do note that there are 5 different types, and they all have different names and different functions. But for purposes of this video, we're going to stick with calling them leukocytes or white blood cells, and they deal with the immune response. So therefore, you can see that blood is a specialized connective tissue. It contains cells in the form that was mentioned as formed elements, and an extracellular matrix, that is the plasma, which their cells are suspended in. And this will conclude our video on connective tissues. They are a very, very diverse group, but they follow a very similar pattern and ultimately serve important functions and purposes to help us sustain life as we know it.